shouldn't affect anything. And then you can start it whenever you're ready, Professor Mohty, when we're at time.
OK, fantastique. OK, bon, là, on peut recommencer à parler. D'accord.
Les choses vont bien en France avec le Covid?
Oui, ça s'améliore.
Ça s'améliore.
Moi, c'est l'impression que j'ai.
On a un peu de problème ici à Alberta, à Calgary. Et, malgré que plus de 80% de la population est vaccinée, on a une grande épidémie now avec le Delta variant. Our ICUs are full. We have no beds in the ICUs now. We have over 260 patients in the ICU. And ce sont les gens qui sont plus jeunes qui la prennent, et malgré qu'ils sont au moins vaccinés pour une dose ou pour 2 doses. Alors, c'est un peu bizarre et effrayant.
Mais ça, c'est la faute aux élections, en fait, que vous avez organisées.
C'est un problème, les politiques ici au Canada. Oh mon Dieu.
Ils sont problématiques partout.
Oh, je disais que j'ai quitté le Liban parce que le Liban était un peu... c'est la politique qui est vraiment corrupte, et ici, c'est pire.
C'est une forme de corruption plus ou moins élégante, en fait.
Oui, c'est ça.
Plus ou moins visible.
Tu l'as bien dit, c'est plus élégant ici. On le fait d'une façon plus élégante.
Oui. Et en France, on le fait de manière démocratique.
Mais au moins en France, ils sont intelligents. Tu entends Macron parler, tu entends c'est chouette les entendre, non? C'est un peu intellectuel. Mais ici, quand tu parles...
Je ne sais pas, Trudeau, je le trouvais sympathique, moi, mais bon.
Oui, il est beau. Si tu as une femme, il est beau à regarder. Il est un très beau bonhomme, mais à parler, mais...
Non, mais j'ai trouvé ses réactions face à Trump, tout ça, j'ai trouvé ça pas mal, mais bon, peut-être c'est qu'une apparence.
Tu sais, ce qu'ils enseignaient à l'université, c'est le drama. Alors, il sait très bien faire le drama. Oui, il parle comme ça. Alors, pour moi, le Canada, c'est une chose de très importante. À le voir, c'est chouette, mais il n'y a rien de matériel intellectuel.
On va voir les élections en France, mais je ne suis pas sûr que ça reste très intellectuel bien longtemps, là.
Eh bien non, mais non, parce que tu te rends pas compte, Cyrille, c'est qu'on n'avait pas réalisé depuis très longtemps que le problème, c'est que ton prénom, tu l'écris avec 2 L-E. Et ça compte, alors que normalement, c'est un seul L.
Ah oui, c'est majeur, ça, le prénom.
Et ça, je pense que ça impacte la prospérité de la République. Moi, Alors, je te parle pas de mon prénom. Alors là, moi, c'est la déchéance totale. Parce qu'on a un futur candidat qui dit que le problème de la France, c'est le choix des prénoms.
J'ai toujours dit ça à mon papa. Nizar, c'est un nom bizarre. On connaît rien de mon nom. On pense que je suis indien, que je suis palestinien, que je suis...
On a de sérieux problèmes avec les prénoms. D'ailleurs, moi, j'arrête pas d'en parler tous les jours chez moi à la maison. Oh là là, orage au désespoir.
Orage au désespoir. Qui a dit ça? C'était le Rodrigue, non? À Chimène. Orage au désespoir, non?
Orage au désespoir, aux vieillesses ennemies.
Aux vieillesses ennemies qui m'attendent, c'est ça, oui. Le Cid, c'est le Cid, non?
Racine.
Racine, le Cid, oui. J'aime ces books. J'aime ces livres.
Ils sont d'actualité, tu vois, 3 ou 4 siècles après. Oui. On le dit, hein. Ils étaient visionnaires.
La dernière fois que j'étais en France, j'ai acheté le château de ma mère, Marcel Pagnol.
Extraordinaire.
J'aime ce bouquin.
Moi, je pleure à la fin, là, les dernières pages, quand il enterre sa mère, quand il enterre son frère, c'est bouleversant.
Oui, moi aussi. J'aime ce bouquin.
Mais tu sais qu'il y a un film, il y a 2 films, La gloire de mon père et les châteaux de ma mère. C'est des vieux films.
Oui.
Et ils sont sur Netflix, hein, Nizar.
Et c'est très bien adapté, c'est vrai.
Ils sont vraiment magnifiques, tu vois. C'est vraiment en plus, ils reproduisent les beaux paysages, l'accent. Enfin, c'est super bien joué.
S'il te plaît, envoyez-moi le nom du film.
Oui.
Ou Cyrille, parce que j'aimerais bien voir.
OK, guys, on va démarrer. Hein. Hi, good evening, good morning, good afternoon, everybody, wherever you are. Welcome to this new journal club by the International Academy for Clinical Hematology. I'm Mohamad Mohty from the Sorbonne University and Saint-Antoine Hospital in Paris, in France. And it is my great pleasure to welcome for today's journal club Professor Nizar Bahlis from Calgary in Canada, and Professor Cyrille Touzeau from the University Hospital of Nantes in Nantes, west of France. So the IACH Journal Club is supported through a generous unrestricted educational grant from Takeda Oncology, and the Scientific Steering Committee of the IACH has chosen for today's Journal Club this recent paper published in the Journal of Clinical Oncology by Dr. Bahlis, and the title, you can read it here, Phase 1 study of Venetoclax plus Daratumumab and Dexamethasone with or without Bortezomib in patient with relapsed refractory multiple myeloma with and without translocation 1114. This is the abstract of this study, and as you can see, this was a phase 1 study in relapsed refractory multiple myeloma. The study included 48 patients, 24 in each part, 1 combining Venetoclax, Daratumumab, Dexamethasone, and the other cohort combining Venetoclax, Daratumumab, Bortezomib, and Dexamethasone. And to make a long story short, these two regimens, Venetoclax-based regimens, produced, according to the authors, a high rate of deep and durable responses in these patients. And you can see here the response rates, but also the MRD negativity rates in those patients treated with one of these two regimens. On the left-hand side, you can appreciate more than 29% of stringent CR, another 29% of CR with Venetoclax, Daratumumab, Dexamethasone, and the overall response rate is more than 91%. And if you look to the MRD negativity, whether you consider the 10-5 or 10-6 threshold, these are really very, very exciting results. And if you look into more details, for instance, into the patient with translocation 1114 versus the patient without translocation 1114, and if you look to the best change in the primary M protein levels, I think if you look on the left-hand side, these blue plots are really exciting, suggesting definitely that this combination is extremely attractive and efficient in this subgroup of patients. So with this and without any further delay, I would like to welcome our panelists for today, Dr. Bahlis, who is the first author of this superb work. Congratulations, Nizar, actually, for this publication, but also Professor Cyrille Touzeau, who, beside being a friend, is a top expert in myeloma and actually has worked a lot on the use of Venetoclax in myeloma, but in hematology in general. So as it is a tradition, my first question is going to be to Cyrille tonight. And Cyrille, I mean, you didn't contribute to this work. So when you saw this paper, what was your reaction? Was it a sort of a wow reaction, or you said, oh, this is another study showing this and this? So please share with us. And here I'd like to remind our audience that this is an interactive activity. Please do not hesitate to send your questions, your comments, your suggestion, whatever you'd like to say. I'll be more than happy to share this with our panelists. So Cyrille, the floor is yours.
Hi, everyone. Thank you, Mohamad, and hello, everyone. Yes, I have to confess that, yes, it was kind of wow reaction. I think this is a very important paper for the treatment of myeloma patient with a T1114 translocation. As you know, there was a first publication 3 to 4 years ago. It was a dose escalation study with Venetoclax single agent in relapsed myeloma patients, and this trial demonstrated a specific efficacy of Venetoclax in patients with T1114 translocation. It was the first time in myeloma, to the best of my knowledge, that a drug demonstrated specific activity in a specific subset of myeloma patients. We read a couple of months ago the phase 3 Bellini trial. It was another step. It was Venetoclax in combination with Bortezomib dexamethasone, and we saw a good efficacy signal in the overall population of relapsed multiple myeloma patients, but also toxicity signal with an overall survival which was better in the control arm. But when we look at the population of patients with T1114 translocation in the Bellini trial, there was a strong efficacy signal of Venetoclax plus Bortezomib dex without a toxicity signal. So now we are there with a drug highly efficient in this subset of patients, but with no approval so far. And this phase 1/2 study is very important because this study confirmed a very, very high efficacy of Venetoclax here plus Dara-Dex in this subset of patients with T1114 translocation. I think it's very, very impressive. It's relapsed patients, 2 prior lines in the T1114 group, and the overall response rate is almost 100%, almost 60% of CR. It's very, very impressive. So I think it's very important there. We have this drug which is very, very efficient in combination with Bortezomib, in combination with Dara. And here we had a cohort of patients, not all patients with T1114 translocation, but with a strong efficacy of Dara plus Bortezomib and Dex. So now we need approval. We need this to treat our patient. And I know that we already treat our relapsed myeloma patient with Venetoclax because, again, we had this strong efficacy, and this phase 1/2 trial confirmed this strong efficacy.
No, thank you very much, Cyrille. So overall, you have really a very positive impression and reaction. And I must confess, I share the same feeling. Also, I'd like to remind our colleagues that I don't think Venetoclax is approved in multiple myeloma, but obviously we are here in a scientific discussion, and we may discuss things that are off-label or not approved in your countries. So I think, Cyrille, you clearly highlighted in this summary several issues. The issue of specificity or maybe precision medicine or targeted therapy in this entity. We'll go back to this. You also alluded to the ideal partner to Venetoclax or combination, and we'll come back also to this, but also you already mentioned the Bellini trial and safety issues, and definitely we have to discuss this. So Nizar, let's start from the beginning, and maybe because you've done an amazing amount of work both in the lab and in the clinic on Venetoclax and other myeloma agents, why specifically the myeloma patient with translocation 1114, why this entity is the most attractive candidate for a Venetoclax-based treatment?
Thank you, Mohamad, for the invitation here, and thank you, Cyrille, as well. So a lot of the work on the role of cell death or the mechanism of cell death in myeloma in general has been done by the group in Nantes with Martina Mio and Cyrille, and I would say Laurence Boyce as well, who's currently in Emory. What they told us for a while that in multiple myeloma, we have a subset of patients or myeloma cells that they are highly dependent on BCL2 for their survival. And just for a little background, if we remember that we have 3 types of protein, well, there's a bit more, but 3 major ones that regulate what we call the mitochondria potential, and that triggers whether the cell will undergo apoptosis or not. And these are BCL2, BCLXL, and MCL1. So the work from Cyrille and Martina Mio and Lawrence Boyes, what they described almost simultaneously, the first paper came from Cyrille actually, that in myeloma cell lines with translocation 11-14, they highly dependent or uniquely dependent on BCL2. In the sense, if you displace this pro-death gene called BIM or BID from BCL or BAX from BCL2, then you trigger cell death. Therefore, the myeloma cells with T11-14 again depend on BCL2 for their survival. And they've shown that eloquently in myeloma cell lines and subsequently in primary patient samples and so forth. And more recently, both the group also showed it's not unique to translocation 11-14. There may be another small subset of myeloma patients without this translocation who have a very high BCL2 to BCL-XL or BCL2 to MCL1 ratio, who also depend on BCL2 for survival. And hence inhibiting that with a small molecule could lead to a myeloma cell death.
So we have a question here. I mean, this is really great. You clearly described the translocation 11-14 as a unique myeloma entity. What is the incidence? We have a question from the audience here.
In newly diagnosed myeloma, it's between 15 to 20%. So it's fairly common in newly diagnosed myeloma. It's actually the most common in newly diagnosed myeloma patients.
We have another question about, because you mentioned that BCL2 is really upregulated here. What about, the question is about the 414 entity. Is this something similar or
no,
this is unique to 11? Okay.
11-14, correct. And it's not really that's, well, it is higher expression, but also you have to think of it in the context of, again, loading. Think of it of 3 empty seats in a theater, in a cinema. You have 3 empty seats, and it depends where this pro-death gene is sitting. Is it sitting on BCL2 or sitting on MCL1 or BCL-XL? And that's what we determines the dependency on which, I guess, pro-survival gene is working in these cell lines. And that's what we refer to often in scientific paper as priming. Where is the priming of these pro-death gene is happening on which protein?
Excellent. So Cyrille, you've done some work, I mean, very elegant work on this. Can you please explain to our audience the rationale for the different combinations that were used by Dr. Bahlis and his colleagues? Because they used Daratumumab, the anti-CD38 monoclonal antibody, and we know that Daratumumab is all over the place. Also, there is now another anti-CD38 antibody in myeloma approved, is Touzeau. So the combination was Daratumumab, and then in the other cohort, in cohort 2, they have added a proteasome inhibitor, the first generation proteasome inhibitor. So what is the rationale of these combinations? And after all, why not IMiDs? Because we know also that IMiDs are very popular in multiple myeloma.
That's an excellent question. First, I have the first partner, which is very common in myeloma, is Dex. And in fact, there is a rationale to combine Venetoclax plus Dex, and it was demonstrated by the team of Larry Boyes in Atlanta. And as Nizar said, Venetoclax works by displacing BIM BCL2 complexes that activates apoptosis. And we know that Dex improves the level of BCL2 BIM complexes. So first, yes, to combine Venetoclax with Dex is probably important. There is also rationale to combine with proteasome inhibitors. And we know that Bortezomib induces the upregulation of another BH3 protein, which is called NOXA. And NOXA antagonizes MCL1. And MCL1 is another anti-apoptotic protein, such as BCL2, which is implied MCL1 in the resistance to Venetoclax. And so with a combination of Venetoclax plus Bortezomib, we kind of target both BCL2 and MCL1. So I have to say that there is not a mitochondrial rationale, to my knowledge, to combine Dara plus Venetoclax, but we target different mechanisms of different pathways. So and I would say probably it's the same with IMiDs. Again, there is no specific action of IMiDs on the mitochondria, but and to the best of my knowledge, there is no clinical trial that evaluated Venetoclax plus IMiDs. But yes, the strong rationale is to combine Venetoclax with Dex and proteasome inhibitors.
Well, this is really great. And so it looks like that on paper, in theory, proteasome inhibitors are really excellent partners in addition to dexamethasone. Also, I must confess, all of us would like to get rid of dexamethasone in multiple myeloma, but that's another story. But then here I have a comment from a colleague, and maybe you can take this, Nizar, suggesting that, well, I mean, I don't know if I'm reading it well. I mean, the comment is about, well, it looks like that adding bortezomib, in the cohort 2 didn't bring much added value compared to cohort 1. Maybe it's not appropriate to compare these two cohorts. I mean, what are your thoughts about this comment, Nizar?
Sure, I will answer this comment, but I'll go back to a prior question you asked about the rationale for daratumumab. It actually came from an observation we made in one of my patients who was progressing on daratumumab Velcade combination, and we realized at that point that she did have T1114 translocation. So we discontinued the Velcade and we added Venetoclax, and she had dramatic response. Even though she had 9 prior line of therapy, she had a basically complete remission lasting for nearly 2.5 years. Which kind of intrigues us about the potential, maybe some synergy. And the hypothesis we raised, what we postulated, that there is probably escape clones that will emerge once you target one specific pathway, as you know, in all malignancies. And here we're targeting BCL2 specifically with Venetoclax. So perhaps daratumumab helps suppress these emerging resistant clones, and hence you get better, longer duration of remission. And the other postulation was made on the rationale of this potential plasticity in myeloma. You can go from a B cell-like to one end to more immature plasma cells in one end. And we're postulating that if you inhibit or the B cell-like with Venetoclax, you may differentiate them more to plasma cells, higher CD38, and daratumumab will take care of that. But I agree with Cyrille, there's no preclinical data in vitro to prove that synergy. We didn't examine that in details. So it's all postulation. Now, going to your question, did Velcade add anything in the cohort we've done? Well, I would say yes, and keep in mind that the cohort where Velcade was tested in combination with daratumumab and Venetoclax was mostly non-1114 patient. So it'll be interesting in the future to examine also a cohort with the 1114 when we add, like in the Bellini, for example, Velcade with daratumumab and Venetoclax. So without a randomized controlled trial, it'd be hard to say that Venetoclax add anything in that control arm. Now, you can argue that the median PFS isn't that different from the Castor trial. Well, I would say slightly better. I mean, 66% PFS at 18 months, that's better than Castor. But again, we need a randomized trial to see how much Velcade contributed in that non-1114 largely cohort.
So looking into these combinations, we have a question from the audience about, and maybe Cyrille, you can take it, about, well, why not using Ixazomib, which is an oral proteasome inhibitor, and this is quite nice, having Venetoclax oral Ixazomib, Daratumumab subcutaneous, so quite convenient, or even Carfilzomib. Do you see any perspective there, Cyrille? I mean,
yes, excellent idea. I would say to combine 2 oral drugs, Ixazomib plus Ven would be probably great. With Carfilzomib, we already have some results. There was a phase 1-2 study, Carfilzomib plus Dex plus Venetoclax in relapsed multiple myeloma patients. And here again, in patients with T1114 translocation, the results were great. I think this study has not been published yet, but it was presented during last international hematology meeting. So yes, Carfilzomib plus Ven should be great, and it has already been done and with great efficacy results, especially in the T1114 translocation patients. And yes, of course, to combine Dara subQ, Ixa, Venetoclax, and Dex in T1114 translocation patient, excellent plan of study.
Okay, wonderful. So we have here, let's move now to some practical questions and details about the use of this drug. And we have one question, Nizar, about the dosage of Venetoclax. I mean, Venetoclax is for me amazing. This is like becoming the Swiss knife in hematology. We use it now extensively in acute myeloid leukemia. We use it in CLL, and it is approved actually. We have really exciting results in myeloma. but then you have a larger spectrum of dosages. So in myeloma, how would you use it from a practical point of view?
So in the current study, as you probably read in the paper, we had 2 doses. We had 400 milligram as initial safety run, and then we bump it up to 800 milligram. So it's slightly higher than the dose is probably used in leukemia or in CLL, and possibly because the BCL2 expression is indeed higher in multiple myeloma. And also from a safety perspective, there is no concern of tumor lysis as we see in leukemia and CLL. So I don't think there's been any cases of tumor lysis in myeloma trials, at least not in this trial. And therefore, there's no need to do this rigorous slow step-up dosing. You can go ahead right away with 400 or 800 milligram. Now, the question, is there a difference in outcome between 400 and 800 milligram? We don't have that data yet. This is being currently studied in this ongoing phase 2 trial, and this will be determined hopefully before we start the phase 3 trial. But I would say for myeloma, the dose is anywhere between 400 to 800 milligram is what the effective dose has been shown or mostly studied.
Excellent. Thank you, Nizar. So it looks like it's relatively straightforward when it comes to the dosage in myeloma. And by the way, guys, please continue to send your questions and your comments. And you can see the panelists are really giving very great, detailed and clear answers. So let's continue with the safety issues and the practicalities. And I will address this question to both of you. Actually, I'd like to get both your point of view. Cyrille, in your introductory comment at the beginning of the journal club, you alluded to the Bellini, the famous Bellini randomized trial. And that was a combination of what I would call the V2 regimen, you know, Venetoclax, Bortezomib, Dexamethasone, and where there was a sort of a safety warning about more infectious complications, which led, unfortunately, to higher mortality in some patients. But then it is very interesting to note that in this protocol, although I think, you know, you've combined it not only with Bortezomib, but also with Daratumumab, there's absolutely no infectious signal, let's say. So what's your interpretation? You know, I'd like to hear from both of you, and because this is important, I think the Bellini trial result has a little bit confused the environment, and now it looks like you're putting everything on track again with this trial, because this is really a very important drug in translocation 11-14. And of course, we don't want to just skip it because of this original signal from the Bellini trial. Cyrille, what do you think?
Yes, I think it was a very negative message and very important. Yes, the result of Bellini was very negative for Venetoclax in multiple myeloma, and it's quite difficult to analyze this safety signal. Yes, there was an excess risk of toxic death in the Venetoclax arm. It was mostly during the first months of treatment, and so there was a lot of debate because sites open for the Bellini trial in an important number of sites. There was no infection prophylaxis as we did in most of our sites, but I think it does not totally explain this excess of risk, because patients in the control arm did not receive any prophylaxis also. So maybe to combine Venetoclax plus Bortezomib will affect T cells, etc. So yes, we have this excess of risk, and based on this, there was, in fact, Bellini could be a trial that led to the approval of Venetoclax, and it was not the case. And the second consequences is that now when we propose a trial with Venetoclax in myeloma, we have to focus on patients with T11-14 translocation. And so in terms of approval, it's much more complicated, because we are waiting for the results of the Canova trial, which compares POM-DEX to Venetoclax-DEX in relapsed multiple myeloma patients. But this trial include only patients with T11-14 translocation. So it's, as Nizar said, it's 15 to 20% of patients. It's important population, but in terms of clinical trial, it's hard to include and to complete the inclusion in this trial. So we are still waiting for a trial that allow approval of Venetoclax in multiple myeloma. So maybe I could stop here. But
so Nizar, how do you explain the excellent safety profile and you didn't encounter any of these infectious complications that were described in the Bellini trial?
So I completely agree with what Cyrille said, and maybe I'll just give it in a different perspective. So if you look at the Bellini trials, and specifically at the T11-14 myeloma, and this is very well detailed in the paper, but also in the supplement, I would encourage whoever read that manuscript to look at the supplement of that paper. There's nearly, I think, 20 supplemental figure and data. So when you look at the T11-14 myeloma in the Bellini, there was no difference in OS, overall survival or progression-free survival. So as Cyrille said, if we're going to focus the development of Venetoclax in T11-14 myeloma, the Bellini trial, even in that cohort, didn't show worse overall survival. And the outcome of these patients is excellent, like we encountered in this study. Now, the other difference, as Cyrille also mentioned, in our study, we implemented prophylactic antibiotic from the beginning. So patients were required to use a prophylactic levofloxacin for the first 3 months, septal prophylaxis, and very aggressive management of neutropenia and infections. So whether it made a difference or not, it's possible. And the third issue, in the Bellini trials, all the death occurred very, very early, which makes me doubt there is any biology driven from the myeloma cells in the immune environment. It's an unfortunate, probably series of events, and they were all infectious related event. But again, even in the Bellini, if you look at the T11-14 subset, overall survival wasn't worse in that subcohort. And the future development, as Cyrille said, we're going to focus on this subset of population, but hopefully slightly larger population, that population with a higher BCL2 to BCLXL or MCL1 ratio.
Excellent. So you really, both of you, insisted on what I would personally call the learning curve, which means, we have learned from prior studies, and by applying some stringent criteria for prophylaxis and management, actually you can keep control of whatever complications. That brings me to one question from the audience, which actually goes beyond this specific article, but I thought it's interesting because, of course, the ICH aims to promote good clinical practice worldwide. It's about antibiotics prophylaxis in multiple myeloma, and the comment or the question from the colleague in the audience was about this randomized trial from the UK a couple or 3 years ago, big oral session at ASH, I think published later, I don't remember exactly which journal, but are you both using systematic prophylaxis when treating multiple myeloma patient in terms of antibiotics, and which antibiotics, if any? Because I think this is like, we are able to do trials with all the fancy new drugs, but we're still not able, I think, in myeloma to establish whether they need cotrimoxazole prophylaxis or not. I may be wrong, but I would like to hear from you because our audience is interested in this. Cyrille?
I use systematically cotrimoxazole in all treated patients. Of course, valaciclovir for zoster, but yes, we systematically use cotrimoxazole. I have to say that we do not use other systematic antibiotic. We do not use quinolone or other antibiotics, but always cotrimoxazole, yes.
What's the practice in Calgary, Nizar?
Very, very similar to Cyrille. We tend to avoid antibacterial beside the cotrimoxazole, or we call it here in Canada Septra, because of the risk of selecting resistant bacteria. But in patient with Venetoclax, at least on this trial, this was required for the first 3 months, as I mentioned. And surprisingly, was very well tolerated, the use of quinolone. We didn't encounter any resistance bacteria or C. difficile colitis with this 3-month. The other thing I forgot to mention, and more recently, I've been more sensitive because of the COVID epidemic or pandemic, the use of intravenous gamma globulin. So if I have somebody with severe hypogammaglobulinemia, I didn't used to practice that, but I'm more cognizant of it, and I'm using it more often right now. Again, I know the data is all over the place about intravenous gamma globulin, but I'm using it especially in patients with severe hypogammaglobulinemia.
Okay, excellent. That was very clear, guys. Thank you. Before moving into the concluding remarks, we'll take one other question, and I think it's a good one. So we usually look into translocation 1114 at diagnosis. Do you recommend to look for it at time of relapse, even if it wasn't present at time of diagnosis? Is there any rationale for this, Cyrille?
Yeah, I think, in fact, the T1114 translocation is quite stable in the disease course of the disease. It's a primary cytogenetic abnormalities, and it remains quite stable. So I think in the daily practice, I would recommend to test the T1114 translocation at time of relapse, if Venetoclax could be an option to treat the patients. At time of diagnosis, if you test it at diagnosis, the patient will keep the abnormality at time of relapse. So yes, maybe to test it at relapse if you consider Venetoclax as a potential drug for the patients.
You have the same approach, Nizar?
Yeah, if it is negative at diagnosis, there's no sense of repeating it unless you have some doubt about the how well the assay was done, if you want to repeat your assay because of technical issues, but not for biological issues.
So the message to our colleagues is that you need to look for it at some point and to know what is the status. Okay. We have a question here, another question about whether this is these combinations that you have tested, Nizar, in this work are relevant in amplification 1Q. Is there any rationale for this?
So I guess whoever asking the question is making the relevance to 1Q does result in upregulation of MCL1. And recently, Larry Boyes looked at the patients who have 1Q gain, if they have an N1114, if their sensitivity to Venetoclax is different. And it really, he had a hard time showing any difference. So even some patient with 1Q gain, they still remain sensitive to Venetoclax, while some were not. So put in other terms, if you have a patient with T1114 and they do have 1Q amplification or gain, I wouldn't exclude them from receiving Venetoclax. Ideally, in the future, we should do what some of the work Cyrille does, and Larry Boyes doing this again, in vitro priming or testing for the sensitivity in vitro to Venetoclax, because it does seem to correlate nearly 100% with the clinical sensitivity as well. I know Cyrille, if you want to comment on that, I'm curious what you think about that.
Yes, I totally agree with you, Nizar. And I think this remark on 1Q amplification and MCL1 points another thing is that could we target MCL1? And recent data are quite disappointing, in fact, because there were several phase 1 trials that tested BH3 mimetics targeting MCL1, and several trials pointed cardiac toxicity, and 2 of these trials has been stopped. So to date, there is no clear view on targeting MCL1. So we have Venetoclax to target BCL2, and there are other potential options to target BCL-XL, and recent data has been published on PROTAC, which are compounds that target specifically and through the proteasome target specific proteins. And so, yes, important remark. MCL1 is important in myeloma survival, but to date we have no way to target MCL1 directly.
This is fantastic, guys. I think we can spend hours and hours debating all this, but my last question to both of you, and let's go for Nizar. Okay, you did this wonderful paper again, congratulations. What's next now?
Yeah, well, I'm very happy that this work is going to form the basis for the planned phase 3 trials. As Cyrille mentioned, there's now 2 phase 3 trials looking to hopefully allow us to have the approval and funding for Venetoclax 1114 myeloma. The first one is the CANOVA trial, which is looking at any relapsed myeloma patients with 1114, comparing Venetoclax Dex versus POM-Dex. But there's another planned phase 3 trial based on this current trial looking at Daratumumab, Venetoclax, Dexamethasone versus Daratumumab, Velcade, Dexamethasone as per the CASTOR in patients with the 1114 myeloma. This study is in the works, currently undergoing review by the FDA. Hopefully, it should be available at multiple sites in the near future.
Cyrille, from your point of view, what's next?
As Nizar said, yes, we need phase 3 trials, positive phase 3 trials to have an approval, and it would be fantastic, especially for patients with T1114 translocation.
Superb. Thank you very much, guys. I'm really very grateful for the time you have dedicated to this journal club, and you shared your great expertise and experience. Our ICH community is really very grateful. Thank you very much. I hope everybody has enjoyed this journal club by the ICH. Our next journal club is in around 15 days as usual, and please stay tuned because I think it's going to be around CAR T cells in autoimmune diseases. I could see this selection yesterday by the steering committee. But also, please don't forget that we have the ICH annual meeting starting in a couple of days on Friday, and you will hear some amazing lectures about state-of-the-art treatment and roundtable discussions in all field of malignant hematology. And of course, as usual, the ICH is a non-profit initiative. Registration is going to be free to everybody, so you just come in and you will enjoy it. So for the time being, thank you very much, Cyrille. Have a good evening, good night, and thank you, Nizar. Have a good day in beautiful Calgary, and thanks to all of you for being loyal to the ICH activities. And wherever you are, please stay safe and keep well, and see you soon. Take care. Bye-bye.
Bye-bye.
Thank you.